Okay, so this one right here is your citric acid cycle. This is that process. So before we had covered this portion of it, notice right here, this is the pyruvate going in as acetyl-CoA here. This is then going to use that energy in those molecules to produce the NADHs and FADH2 along with a single GTP. Okay, so reaction 1, the first reaction, this is going to be formation of citrate. Now notice this is the naming citric acid cycle. This is the one it's named after. Okay. The acetyl group, this is the acetyl-CoA, which is 2 carbons. It's going to combine with oxaloacetate, which is the 4 carbons. This is going to form your citrate, which is 6 carbons long. And you'll also get CoA back. Okay, so your coenzyme A regenerates. Step 2 is going to be the isomerization of isocitrate. You may actually notice that both say iso, right? Because they're isomers. Isomerization, making an isomer of citrate. Okay. And so what's going to happen here is the OH and one of the hydrogen atoms, they're going to be swapped. And so instead of citrate, you're going to have the isocitrate rearrangement. This rearrangement is actually incredibly important for this because that isocitrate needs to be oxidized on the next step of the reaction. The current location of the OH will not allow that oxidation. And so by switching them, you allow this next step's oxidation. So this is going to be step 3, reaction 3, which is going to be the first oxidation, and decarboxylation, which is that release of the CO2. And so this will be your first release of CO2 in this pathway. And so this is going to be an oxidation. It's going to take 2 hydrogens across that carbon-oxygen single bond away, and that's going to give you the ketone. That ketone there is called alpha-ketoglutarate. This is an oxidation step, so you're going to have something that's reduced. And so NAD plus is going to be reduced to NADH. I know, did you ever think you'd be able to understand that sentence?
Makes my head hurt to understand that sentence, actually. I'm like, wait, let me think about this again.
Yes, but you do. Yeah. Okay, and so you're going to accept that proton electrons are going to be removed during the oxidation. The 6-carbon isocitrate will be decarboxylated. That's going to give you now a 5-carbon, and that's the alpha-ketoglutarate right here. You may notice that a lot of these have keto in them. You can actually start making connections in your head if you'd like. They are. Reaction 4, the second oxidative decarboxylation. This is that second one. Your thiol group, which is your coenzyme A, is going to be oxidized. It's going to lose a hydrogen. Another NAD plus is going to be reduced to NADH. And this is going to give you alpha-ketoglutarate, which is 5 carbons. You're going to have that decarboxylized into the succinyl group. This succinyl group is actually going to be attached to a coenzyme A. That's why it's the succinyl instead of like succinate. Okay. And so that coenzyme A will be bonded to the succinyl group, and you'll have succinyl-CoA that you get out of this. And notice again, this is another step where you're going to reduce that NAD plus to NADH. And so you're harvesting that energy in the form of NADH. Step 5 is going to be the hydrolysis of succinyl-CoA. So succinyl-CoA is going to undergo a hydrolysis. This is going to give you succinate now, because we're splitting it off, and a free coenzyme A again. So we're going to free that up again. The energy that's produced is going to give you the high-energy nucleotide guanosine triphosphate, which is GTP. And you'll get that from GDP and P prime, free inorganic phosphate. This GTP, by the way, is energetically equivalent to an ATP. It can be converted into your cell, and your cell does that quite a few times because you need various amounts of GTP, ATP, CTP, and UTP to make your RNAs. The D, small d in front of them, except for U goes to T, obviously, for the DNAs, right? And so you're constantly having to swap back and forth with these. Okay? So you have enough of each of those to make your DNA and RNA. Reaction 6 is going to be the dehydrogenation of succinate. You may recognize this reaction as the same thing that happens to peanut butter. And you do it at reaction 6. You also do a dehydrogenation. Ooh, so we're doing some dehydrogenation just like we did back in the organic chemistry reactions. I know you thought that organic chemistry section was like, why does Dr. Clausen teach us this? This is why. Because all of those reactions actually take place in your body. So this is going to be taking hydrogens off and giving you a double bond. So one hydrogen is eliminated from each of the 2 central carbons of succinate. This gives you a trans double bond here. And so this is going to produce fumarate. These are going to be the 2 hydrogens are going to give us the coenzyme FAD going to FADH2. And so notice you've again done another reduction. Okay. This is the oxidation portion right here with the fumarate giving us the oxidized species. Less hydrogen bonds, right? And then the FADH2, more hydrogen bonds, so it's the reduced form. Now, reaction 7, this is going to be the hydration of fumarate. I know, another reaction you probably recognize. So this is adding water across something, isn't it? So we have the double bond, we add a water across it, an OH and an H, and this is going to give us malate. Okay? Now, this is actually really important because this sets us up for our next step, which will reset all this. So now we have an alcohol. So we can do another oxidation of that malate, and that's reaction 8. And so our secondary alcohol that's in malate that we just set up, right? We're going to take a hydrogen off of that OH and off of the carbon next to it. And that's going to give us our ketone. And so that ketone is called oxaloacetate. And it's going to provide the protons and electrons to reduce another coenzyme NAD+ to NADH, which is another harvesting of energy. Okay. And so that's reaction 8. Notice at the end of this, we have regenerated oxaloacetate. What did we start with again? Oxaloacetate. So we are back to the beginning of our cycle. We have reset. Now we can accept another new acetyl-CoA. Okay. And keep on chugging along. Exactly, hence the cycle. And so you recycle 4 carbons out of 6 carbons every time. 2 carbons always get freed as CO2s. All right, so the succinate is a symmetric compound. We can no longer track the acetyl group that entered the cycle as acetyl-CoA. So when we get to that succinate over here, because it's symmetric like this, we no longer actually have an idea of which carbons we have coming out. And in fact, you can track them by labeling them isotopically. And when they do that, it basically ends up a split. You get like a quarter reduction every time. And it's because one of these you can track right here. And so it's the old one. The other one you can't track, and so you have a 50-50 chance. So, yeah. Now, let's do a summary of this. So what's the overall results of all that? Okay, so one turn of the citric acid cycle. This is going to produce net energy output that is 3 NADHs, and then we have 1 FADH2, and then we have 1 GTP, which is energetically equivalent to an ATP. You can just think of it like an ATP, okay, in terms of the energy you're producing. You'd produce 2 CO2s, and you get 1 coenzyme A back out. So your coenzyme A that started attached to an acyl, we get that back. That doesn't get consumed, and so you can reuse that to get another acyl back in here and do it again and again and again. Okay. Now, this is that net reaction again. And so we had the acetyl-CoA plus 3 NAD pluses plus an FAD, a GDP, a free phosphate, and 2 waters. They're going to give us 2 carbon dioxides, 3 NADHs, 2 H pluses, an FADH2, a coenzyme A, and a GTP. It's like a partridge in a pear tree, right?
Yeah.
I should get like a Gregorian chant going.
That.
Yes, best type of chant. All right, so 2 ATPs are going to be produced in the glycolysis. 2 ATPs are produced in the citric acid cycle. Where is all the energy actually coming from then? That doesn't sound like a lot, does it? You did all this work and you got 4 ATPs. Yeah, that's not great. All right, so that's the NADHs and FADH2s, right? That's where you're going to get your rest of your energy from. So remember, in glycolysis, you had 2 NADHs per glucose that were formed. The pyruvate oxidation to acetyl-CoA is going to be 2 NADHs per glucose that you produce there. 1 per each of the pyruvates. In the citric acid cycle, we're going to have 6 NADHs and 2 FADH2s per glucose. Again, that's 2 cycles of the citric acid cycle. Okay? And so the high energy reduced forms of your nucleotides are going to transfer energy and hydrogens through the inner mitochondrial membrane. This is going to be where you form your water, and this is actually going to be where you're going to end up using the oxygen. And so this is going to be your electron transport, which will build up charge, just like a battery does, so then it can drive the oxidative phosphorylation. The oxidative phosphorylation will be the process of actually fixing that energy into ATP. Okay? And I want to point out, I'm not going to have the videos in here. I should put them in again, but people kind of got lost when I did the videos because they're, by necessity, more detailed than this course is going to go into. If you actually look at how these things work, it looks exactly like an electric vehicle or like an electric factory. Okay? You charge up a battery, literally a battery, where you concentrate ions on one side, and that energy difference of those ions being concentrated drives processes. Okay? And then you take that energy, you drive a crankshaft, and it moves around this little press that literally is going to take ADP and a free phosphate and go thunk. Okay? It is quite literally like you've seen in how they make things, factory floor, where it has the press, thunk, 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 joining the things together, right? It is shockingly like a machine, okay? And they've done studies where they've actually labeled these so you can see the little crankshaft going. And it's really funny because it looks exactly like a factory. Dunk, dunk, dunk. Yeah. So, again, the energy generated as a result of that process, that's going to be used to drive the reaction of ADP going to ATP, which is how you'll then fix that energy in ATP. That process where you fix the energy from ADP to ATP, that's oxidative phosphorylation. Notice phosphorylation meaning to put on a phosphate, and it does it oxidatively. Okay? As in redox reaction, right? Oxidation reduction. So let's talk about the electron transport real quick. We have the mitochondria. These are going to be ATP factories of the cell, if you will. Okay? Quite literally. This is going to reduce nucleotides from your citric acid cycle. They're going to be produced there. The energy upon oxidation is then going to be used to generate ATP. One of the things I'd like to point out here is you're thinking of these probably right now as separate things. Like they happen in separate places and they're not interlinked. Okay? What you're going to notice when I start saying these names, they are not separate. They're the same sets of enzymes and complexes feeding right into each other. Okay? This is like your car. It's not like two things hooked on a wire and then they're like in different areas of the country. Okay? Or different parts of the cell. This is like two gears moving each other. They're very interlaced. Right? There is no actual separation. Okay. So, the reactions for your citric acid cycle are going to occur in the matrix of the mitochondria. The NADH and FADH2 begin their journey in the inner membrane here. The enzyme complexes, which we're going to talk about 1 through 4 first for the electron transport chain. And then we'll talk about enzyme complex 5, which will then be the oxidative phosphorylation. Okay? These are going to be embedded in that inner membrane of the mitochondria, usually on those cristae, which are those little points that sort of jut out into it. And they're going to contain a set of electron carriers. These are going to transport the electrons, which will then pull the protons through that membrane. And those are going to be the electrons and protons we were talking about moving onto the NADH and FADH2s. Remember how we kept saying like NADH and H+? That's that. That's that proton. Okay? And our FADH2, of course. So, these are going to move them through the inner mitochondrial membrane. Remember, the protons can't just move through a membrane, right? Because they're positively charged. The membrane is going to be nonpolar, right? It's not going to allow positive or negative charges through it. So what you're going to do is you're going to pump it all into one side. You're going to get this really high concentration of H+, meaning it's very acidic, right? So it's an acid battery. It's literally an acid battery. Okay? And then you're going to have that... did anybody ever feel where I mixed up concentrated acids? Oh, I didn't get a chance to do that with y'all? That's a shame. Probably safer, but it's a shame. So if you take a concentrated acid and then put it at a lower concentration, you dilute it, what you feel is that radiates a bunch of heat out. Okay? And so that's because it's energetically more favorable to be diluted. Because you're going to lower that charge, that positive charge, right? And so what's happening is you're building up this positive charge on one side of that membrane, and then the complex 5 is going to let that positive charge through, and that's going to drive your crankshaft. Kind of like you built up a bunch of like hydraulic pressure that then drove a crankshaft, right? See how I'm saying? It's literally, it is quite literally factory. Okay? All the things that you would have learned about a factory, you can apply those mostly. Big difference being here is this is a chemiosmotic rather than just normal osmosis. Because it's positive charge, right? Rather than like pressure of water. Okay. So, again, we build up that charge. You're going to have 2 electron carriers. You're going to have a coenzyme Q and a cytochrome C. These are coenzyme Q and cytochrome C are not going to be firmly attached to any one complex. Their purpose is actually to shuttle the electrons between the complexes, so you can get the most efficient use out of those as possible. Another way to think about this is, like if you were just to uncontrollably move around electrons like you do in a wire, that heats it up, doesn't it? And you don't want to get heated up. You'd burn up. You'd have problems maintaining your temperature, right? And so you can use this to generate a bit of heat, but you don't normally want that. You have enough difficulty cooling yourself as is, right? And so normally this does it as efficiently as possible, meaning we need to recycle as much energy as possible and not allow that to go to waste, which is heat. Okay, so this right here is the electron transport chain. And remember, this is going to be directly joined in. You'll notice that when you see the names. Okay. So this right here is the matrix right here that we're getting all these things from the citric acid cycle. Okay. This here is the intermembrane space. And so this is where you're going to build up that positive proton charge. Okay. And so we're just going to follow the electrons for now. We'll worry about the protons afterwards. Okay. These electrons are going to pull protons through, but we'll worry about that after we figure out the electrons. Got to follow these first. So what you're going to see is you're going to have NADH going to NAD+. And this is going to give an electron and a proton to complex 1. You're going to have your Q, coenzyme Q here, shuttle that to complex 3. You also then have complex 2, which is going to use FADH2 going to FAD to put a proton and electron onto Q. This is going to get shuttled to complex 3. Now, these Qs don't need to be the same Q. Okay? You can have multiple Qs. There's just going to be a free flow of Qs moving these things around. If you've ever seen a factory, they usually have like forklifts, then they move the pallet, go to the next one, right? Yeah. This is the forklift person running these things between the two lines. Okay? It needs to be neutral because this right here is a membrane, isn't it? And this is a non-polar environment. So if it's charged, you wouldn't be able to be in there. Okay? Okay, complex 3 then is going to use these Qs here, and it's going to be transferring those electrons off onto cytochrome C. Now, it's accomplishing more than just transferring the electrons. It's pumping protons through. But for now, we just need to concentrate on where do our electrons go. So this is going to transfer those through a pretty complex series of little metal ions that they jump to. It's kind of a cool thing, actually, but it's a very in-depth thing, and it'll take some time, so we won't be able to get through all that. This is going to transfer this off onto cytochrome C. Cytochrome C, again, is going to be neutral, so it can move around the shell membrane. And that's going to transfer that to complex 4. Complex 4 is where you get the big payoff at the end, okay? And so this is where you finally use those oxygens. You're probably like, where the heck did the oxygens get used, Dr. Clausen? You've made CO2, you've taken all these things apart, you made the NADHs. We haven't used any oxygen yet, and we finally get to use the oxygen. Okay? And so this is that breathing you're doing in. This is where those oxygens are going, or at least the vast majority of them. So this is going to take 2 H pluses, 2 electrons, and it'll use half of an O2 each time it does that. Okay? 2 of these would use an entire O2. And they're going to give you water then. Right. And so notice that our CO2 production is actually uncoupled from our oxygen consumption, which is very, very efficient. It keeps us from producing a lot of heat energy. Notice I don't have complex 5 in here yet. That's going to use the H pluses you're going to pump into here to power that next process. That's going to be oxidative phosphorylation. Okay, so let's go through those complexes in a little more detail, a little finer comb, okay? So, complex 1, you're going to have the NADH. It's going to enter that electron transport. Okay, this is the process for one NADH. Notice we've made more than one NADH. Okay? This is going to take and oxidize the NADH. You're actually going to have 2 electrons and 2 protons that are going to be transferred to an electron transporter coenzyme, which is called Q typically. Okay? And that's this one right here. That's because of the quinone right here. This is ubiquinone, and these are the quinone. Okay? This is going to be transferred to that coenzyme Q. It's going to reduce these 2 ketone groups right here to alcohols, which is hydroquinone. And typically that's just known as QH2 because that's just easier. Okay? And that's what that looks like right there. Now, notice that that right there is the reduced form, isn't it? This up here is the oxidized form. Okay, this is the higher energy form.
Is it something like this? What may, like, not club soda, but the other one that's similar to club soda has, maybe it's like quinine in it, and it'll glow under a black light?
I'd have to look. I don't drink a lot of club soda
It's not
stuff.
club soda, it's the tonic water.
Oh.
Tonic
Well, we can
water,
look at it
the
sometime.
difference between like tonic water and club soda is it has like quinine? I don't even know. So I just heard it.
Mm.
And then you can make it glow on like Halloween.
Oh.
I've never actually read what's in it though.
Yeah.
I was like, is this what's in it?
Yeah, we could look sometime.
Does this glow under a black light?
I am not sure. Probably. Many organic compounds that are biological in nature glow under black lights.
There we go.
Yeah. But that's why I like urine and
Yeah.
other things. in your hotel room.
Uh-huh.
Yes.
Yeah, but-
Yes. But yeah, I would. This probably would glow in a black light.
Oh, yeah.
In fact, this structure right here tells me it would like actually be pretty fluorescent.
There
Yeah.
you go.
But it could not be related. It could be related. I don't.
Mm.
Yeah. I just don't drink any of that stuff, so I have no
me
idea.
either.
Okay. So, anyways, this is going to have your NAD plus regenerate. By the way, notice that because we've regenerated this, we can return it to the catabolic pathway, and so we can put it back in the citric acid cycle. If we needed it back over, you could transfer it out of the mitochondria and put it into the glycolysis. So we've regenerated these NAD 8 pluses.
Those are quinine. No.
Quinine.
So that's
So it's a related compound.
Yeah.
Yeah, it's named after this structure.
Yep.
But I would have to look at it to see.
Okay.
It probably doesn't have all this on.
I didn't say. And the molecular structure.
Oh, you should.
We'll do that later.
So the overall reaction for complex 1 is NADH plus H plus Q gives you NAD plus QH2. Okay. All right, so complex 2. Notice the names here. This is NADH dehydrogenase. This one right here is succinate dehydrogenase. It is directly linked into that succinate step. Okay? It's the same enzyme, actually. All right. Now, the FADH2 is going to enter your electron transport chain. This is after the reduced nucleotide is produced in the conversion of succinate to fumarate in that citric acid cycle. And so the 2 electrons and 2 protons from your FADH2 are going to be transferred to coenzyme Q. This is going to give you QH2. And so this is going to be FADH2 plus Q gives you FAD plus QH2. So we've generated another QH2. Okay, so these QH2s, their payoffs are going to happen in complex 3 and 4 as well, because 4 is going to work with 3. And so at complex 3, you're going to have your reduced coenzyme Q, that QH2 we've been talking about, generating each of these times. These are going to be reoxidized back to ubiquinone, which is Q. The electrons are going to pass through a series of electron acceptors. It's actually kind of a cool thing. Anybody ever seen Frogger? Where it has the frog jumping from lily pad to lily pad?
Mm-hmm.
Okay. It kind of looks like that. So you have these little metal ions that you're going to have the electrons jump onto, and one after another, like a little platform. And so it's kind of a neat way to do it. Keep in mind, we don't have wires in us, right? So we can't, like, transfer electrons.
Mm-hmm.
It's pretty complex transferring electrons in a biological organism. Okay? It's not as easy as you might think. Now, these electrons are going to pass through then that series of electron acceptors. They're going to arrive at cytochrome C. This is going to move those electrons from complex 3 to complex 4. Okay? Now, again, remember that when we're talking about these things, we're going to move hydrogen ions through at complexes 1, 3, and 4. Okay? And so that's where we're getting those payoffs. And we're going to add that in once we've talked about the electrons going through. This works very, very similarly to how, like, your electric motors work in an electric-driven vehicle of some sort. A drone, electric race car, or an actual car. So it actually has the electrons moving. These electrons are going to be negatively charged, right? So they're going to move through, and they're actually going to pull the protons with it. And so how an electric vehicle works is you have a magnet, and then you have an electromagnet that's going to move a charge, which then the actual magnet follows. And that's how you actually use an electric vehicle. Okay? So it's very, very similar concept, actually, which is quite amusing, isn't it? Yeah. We work so much like the stuff around us now. Okay. So we have made cytochrome C reductase. At that cytochrome C reductase, we have made that cytochrome C in the reduced form now. Okay? That cytochrome C in the reduced form is going to go into complex 4, which is cytochrome C oxidase. And this is going to be where we take single electrons from cytochrome C. We're going to go through another set of electron acceptors. This is then going to combine with hydrogen atoms and our oxygen, and that's going to form our water. And so the overall reaction and the final step for those electrons, we finally used them all up, is H2 plus, two H2 pluses, plus 2 electrons, gives us 1/2 an oxygen, and that's going to give us an H2O. Okay? So this is again where we finally use up that oxygen, and we're finally using the last of that energy from those electrons. Okay? Now, oxidative phosphorylation, remember this is where you're actually going to make ATP. So the chemiosmotic model is going to link our electron transport to the generation of our proton gradient. And this is going to be across that inner membrane, and this is going to result in that production of ATP. So we build up the charge in our battery, which is that membrane space, right? And then we're going to use the force of that charge wanting to escape to drive complex 5 oxidative phosphorylation to make ATP. Okay?
A little turbine.
It is. It is little.
Absolutely effective.
Yeah, it's an actual turbine. Literally moving along just like it would have. Driving a little press.
Right?
Yeah.
Okay.
Yeah. In fact, has anybody ever seen a water wheel?
Yep.
Yeah. So if you've seen the old style water wheels that do the presses, they used to use that for like forming metal.
Mm-hmm.
It's yeah. It is exactly like that. Just instead of water pressure, you're driving with H plus pressure.
Do you think we figured that out on our own, or was it biologists looking at stuff? Because that to me predates like microscopes.
Yeah, we figured that on our own.
That's just crazy.
Yeah,
so
we
crazy.
did, and we figured out H plus batteries on our own too.
Right.
Yeah, so acid
Also
batteries,
crazy.
just like this process, and it's just because it's just an efficient way of doing it. Yeah, and so
Crazy.
yeah, you can come up with it from different angles. It's like crabification.
It's like, what?
You guys have never heard of crabification? Okay, so crab-like organisms have evolved at least 11 times on the planet separately. And so species all the time are going to crab-like forms.
Like a hermit crab versus soft-shell crab versus...
Yeah, so a bunch of the things that people call crabs are not actually crabs.
Like a hermit crab.
Yeah, they're other organisms that evolved into crab-like forms. And it's just because that form is very, very, very good at surviving in a fair number of niches. Yeah. And so you get like constant evolution towards that form. Yeah. Which is really weird, right? Now, interestingly enough, you may not know this, but hominids actually had a bunch of related species at one point. We're the result of a mixture of those different hominids.
You mean like Neanderthal versus Homo sapiens?
Yeah, there was at least 5. Yeah, most of them mostly died off, but there's genetic parts from all of them in the human DNA.
Mm-hmm.
So, yeah, we're a mix.
We're a hybrid.
We are. So, in these models, of course, we have the 3 complexes, complex 1, 3, and 4. Those are the ones that are going to span the inner membrane. Those are the ones that pump the protons across out of the matrix into that inner membrane space. So they're going to be where you move the protons across. Complex 2 does not move protons in and of itself. As you're going to notice later, this makes FADH2 less efficient at producing energy. Okay? At least ATP. The difference in charge and concentration of protons on either side of the membrane is going to give us what's called an electrochemical gradient. This is the same thing that charges up batteries. Okay? And so you build up an electrochemical gradient, which is what's going to power that complex 5 eventually. This is going to be where you're going to take those protons, you're going to move them back into the matrix through that protein complex. This is going to be called complex 5 or ATP synthase. ATP synthase, synthesizing ATP, right? Yeah. This is going to be a resulting release of energy, and it will drive the synthesis of ATP. And so this is what that looks all together with the proton amounts in it. So that complex 1, when we are moving those H pluses through, is going to move 4 H pluses. Complex 2, none, because it doesn't span the membrane, right? Can't move any H pluses. Complex 3 is going to move another 4 H pluses. Okay? So notice that NADH ends up moving a lot more H pluses, actually, doesn't it? And then complex 4 is going to remove a further 2 H pluses through. Okay? ATP is going to be generated. 4 H pluses is going to give you an ATP. Now, these numbers are a bit approximate. This actual numbers actually change depending on the individual and the organism. Different organisms tend to have different efficiencies for this, and different people can actually have slightly different efficiencies.
What about all over your body? Is it like, I'll always think 36, 34, 32, or is it like over here, or sometimes?
It is possible you could end up with slightly different efficiencies depending on your part of your body, and especially age.
Yeah.
Would be your big one.
Or even metabolic health around those cells.
Yep. Oh yeah, that's going to play a bit. If you have metabolic issues, you're going to play a big one.
Yeah.
This also, this efficiency of the citric acid cycle and then the electron transport chain and then oxidative phosphorylation on top of that is a large part of the chemistry that like balances your mood and energetic feeling. So it's actually kind of shocking how much of this. Now, this gets kind of complex because it's not, your body doesn't respond to things like you probably think it does. Like, if you're tired, one of the best things you can do is exercise. Yeah.
Yep, not eat, but that's what people do.
Yeah. You can get a very minor alleviation of tiredness by eating sugar, but it will wear off very quickly.
So.
Yeah. And so it's actually a worse thing to do. Yeah. And did you guys see that rapper recently? Those in the news? Okay, there's a French rapper that did like a event where he ran for 24 hours straight, and then he did a music video right after. Yeah. And you can run for 24 hours straight, like actually run for 24 hours straight, and then not be tired. Okay? It all kind of depends on your oxygen level intake. If you exceed into the anaerobic versus aerobic. Yeah.
And if your body can get into hypnosis easily and start burning off some of your excessive fuel, because 24 hours in, you've wiped out your glucose stores.
Well, I think he probably was maintaining food in. I didn't really watch it. I don't care that much about the rapper or anything, but yeah, people usually don't. He's the first person to ever run in 24 hours under observation. So kind of an interesting thing, and they did it with the medical team and all that, so they got the data.
The military does that. The military will put them on marches for over 24 hours.
No, I mean straight. No breaks at all.
Yeah?
Yeah, they put them on marches for 24 hours, but they have resting states that they can achieve during the marches. So it's not an outright run for 24 hours.
I've been told that they don't get stuff. They're trying to see who's going to go epileptic by stressing them out physically and tired. Like, I don't get to sleep. I don't get to stop. Who's going to have an epileptic seizure? We'll put them through it for 3 days.
Yes, that is true. No rest. No full rest, no sleep. But typically they do have some rest periods in there. Yeah. They don't full march for 24 hours usually. I've never heard of that before. Yeah.
Anyway, go ahead.
So, the electron transport and ATP synthesis is going to be uncoupled. You can do one without the other. So if you uncouple them, they can be done one without the other. Not so much necessarily you. Okay. If you uncouple them, the energy that would have been harnessed in ATP will actually be generated as waste energy. Waste energy is by definition heat. Okay. And so, by the way, a heater in your house, you know how they go out to the Walmart or whatever store and you go, it goes like 100% efficiency, high efficiency heater. All heaters are 100% efficiency because any of the waste they make is in the form of heat. And so it's impossible for them to be any less than 100%.
Some of them glow.
Again,
I guess we could technically say that I don't need my heater glowing. That would be an energy.
Ultimately, unless that light escapes your house, that's also going to be heat.
Fair.
So the blowing it, unless you let it escape your house and blow outside your house, it is gonna be heat. Yep. It's all heat. Yeah. So the generation of heat in your body is actually called thermogenesis, and this is how you can actually correctly produce heat. Okay. This is one of the reasons why you can actually get people to burn more calories by just doing things in cold environments. Okay. Mm-hmm. Probably a lot of why people up in Northern Europe are so great in shape. They have like holidays where they go out in the freezing cold and just run and then swim across lakes. So, it's probably a great idea. Some animals adapt to cold climates by producing thermogenin. It's a protein that will actually work to uncouple the electron transport and oxidative phosphorylation. And so will allow protons to leak back into the matrix. And this will then allow that to generate heat because it's going to be generating waste heat. Same as if you did a concentrated acid and diluted it. So animals that have this are things like bears. They're going to have higher amounts of tissue that's called brown fat. And so newborn babies actually have higher levels of brown fat as well than adults. And this is because, is everybody aware of the link of volume versus heat loss? Okay. So surface area increases slower than volume. And so the more surface area you have, the more heat loss you have. So if you have more volume, by definition, you're going to tend to have less heat loss. Okay. And so the greater volume, the less heat loss in proportion to their volume. So babies actually lose heat very quickly, very easily. And so this is actually why to maintain their body temperature, they have more brown fat, they're more chubby, and they're going to have brown fat deposits that are going to be located near major blood vessels. This is to help warm up that warm blood through the body, and so it can circulate and keep everything warm. They're going to tend to generate more heat to warm the body's surface, and that's why, like, if you hold a baby, it's warmer than you. It feels warm. Yeah. That baby literally is warm. It maintains a little bit warmer of a temperature. It loses heat easier, though, too. So ATP production, let's start counting up and do our count up. We have glycolysis. That's the oxidation of the glucose. It's going to produce 2 NADH molecules, 2 ATP molecules net. The glycolysis is going to occur in the cytosol. Your electron transport is going to draw the NADH from the matrix inside the mitochondria. And so we're going to transfer those over to the mitochondrion to generate energy. 2 NADHs from glycolysis can be shuttled into the matrix, enter the electron transport chain. This is going to give us 5 ATP. So you get about 2.5 ATP per NADH. Okay. Oxidative pyruvate, oxidation of pyruvate. This is going to be after glycolysis. We have the pyruvates, the 2 pyruvates. They're going to enter the mitochondria. They're again then going to produce 2 acetyl-CoAs, 2 CO2s, and 2 NADHs. And so that oxidation of those 2 pyruvates is going to give us again a net of 5 ATP. Because you do it twice, 2 times 2.5. Citric acid cycle, you're going to have 2 acetyl-CoAs that are produced and going into the citric acid cycle. Each of these citric acetyl-CoAs cycles of the citric acid cycle is going to give us 2 CO2s, 3 NADHs, 1 FADH2, and then 1 GTP, which is equivalent to an ATP. Again, you have 2 of these, so you're going to do 2 cycles of that. So we double those numbers. And what we see is we get 6 NADHs producing 15 ATP, 2 FADHs, 3 ATP, and then the direct results of those 2 GTPs being 2 ATPs. So the total for our 2 acetyl-CoAs in the citric acid cycle, that is 20 ATP worth of energy. And so if we add all of it together, we're going to get approximately 32 ATP. Okay? This can range from 30 to 34 depending on the organism. But what I want you to remember is the 32 ATP. That's your normal numbers. Okay? Okay, so this is actually that full sum of that pathway. Right here is the cytoplasm. Right here's our mitochondria. There's your energy outputs and your different outputs for all these, and your various different inputs. So let's talk about fatty acids. So glycogen and glucose can become short and not available. And so cells can then oxidize fatty acids. This will produce acetyl-CoA through what's called beta oxidation. And so that is the Greek symbol beta. Carbons are going to move 2 at a time from an activated fatty acid. That activated fatty acid is joined to coenzyme A. And so that's an activated fatty acid. So we have here an activated fatty acid. This is going to be called a fatty acyl-CoA. And the reason this is called a beta oxidation, this right here is our acyl group. See that? And this is going to be the alpha carbon. Next one over, as in the Greek letter alpha, first letter. And then we have our beta carbon, which is the next one over after that. And so we're actually going to oxidize and break the bond between the alpha and beta carbons. And we're just going to do that over and over and over. And that's going to cut off an acyl-CoA each time. Okay. So that beta oxidation is going to include a 4-reaction pathway that converts our CH2 of the beta carbon to a beta ketone so that we can reset this cycle. And so once the ketone is formed, your 2-carbon acetyl group is going to split from your fatty acyl carbon chain. 1 cycle of the beta oxidation will give you 1 acetyl-CoA. It will also give you an FADH2 and 1 NADH. Notice that's actually a pretty good payoff every time you're generating one of these, right? And so what you'll notice is fatty acids are worth 9 calories, right? Normally like a protein or carbohydrates is worth 4. And so the fatty acids are quite a bit more energy dense. That's why we store our energy in fats. Each cycle is going to shorten your fatty acid-CoA by 2 carbons. This is actually the beta oxidation cycle. It will be a cycle, but we will cut that fatty acid by 2 carbons each time. Okay? And so reaction 1, you have oxidation. This is going to be another dehydrogenation, same as we have been talking about before with dehydrogenation. Remove 2 hydrogens across a bond to make a double bond. So this first reaction is going to remove 1 hydrogen from the alpha, 1 from the beta carbon, and this is going to form a double bond between the alpha and beta carbons. We're doing an oxidation, so we're going to reduce FAD to FADH2. That's going to be the reduced species. The next step is going to be hydration. So we're going to add water across that double bond. And so that's going to add an OH and an H across the alpha and beta carbon double bond. The OH goes on the beta carbon. And so we're putting an oxygen there so we can do our next step. The next step is going to be another oxidation, another dehydrogenation. We're going to remove a hydrogen off that OH and a hydrogen off the beta carbon, and that's going to give us a ketone. And so now we've generated our ketone. We've done an oxidation, and so we're going to reduce NAD plus to NADH. Notice we're harvesting energy. The next step is reaction 4. This is going to be our reset step. And finally, we're going to cleave that acetyl-CoA. And so we're going to remove that acetyl-CoA off the front the head. And this is going to break that bond between the alpha and beta carbons. We're going to have a second CoA that comes in, and it's going to attach to that beta carbon, which is going to cause that bond to break. So it's going to cause the alpha-beta carbon bond to break. And so this is going to be that fatty acyl-CoA that's then released from the acyl-CoA. And that's going to give our two species. Right here we have some pictures to make this hopefully a little bit clearer. And so what we see here is our fatty acyl-CoA, right, with 18 carbons. Okay. We do that first oxidation here to get a double bond. The hydration right here to get the OH. Then we do the next oxidation right here, and we get our double bond on the carbon oxygen. This is then going to be attacked right here by another coenzyme A. And so we get another coenzyme A attacking here. And that's going to free the acyl-CoA and give us our fatty acid acyl-CoA, but 16 carbons off. See, we've cut 2 carbons off. And we're going to load that back in, do that again, and again, and again, until we're left with just these, right? Acyl-CoAs. Okay. So if we do these cuts, if we go ahead and do these beta oxidations, what we're going to notice is we're going to do this 8 times if we have 18 carbons. And the reason we do it 8 times is we're going to produce 9 acyl-CoAs, right? Now that last acyl-CoA, though, does not require a beta oxidation to happen, does it? Because it's already 2 carbons. There's no beta. You can't go any further. And so what you're going to see is you always take your number of carbons, you divide it by 2, and that's your number of acyl-CoAs. And then you would look and go -1, and that's the number of beta oxidations you would do. Okay. Now, the total number of ATP produced from the NADH, FADH2, and the acyl-CoA generated is going to be based on that NADH, FADH2, and then our citric acid cycle. Let's talk about what happens if we have too much acetyl-CoA, though. And so this is called ketosis. So in the absence of carbohydrates, the body's going to break down body fat through beta oxidation. This will continue ATP production. The liver's going to produce AA glucose from the pyruvate. This is going to be done through gluconeogenesis. It's for our use in our tissues that require glucose to power them, which are going to be things like our brain and our heart. Our brain can only use glucose to power itself, essentially, and so we need to produce it all the time for our brain. The oxidation of large amounts of fatty acid is going to cause acetyl-CoA to accumulate in the liver. When this occurs, your 2-carbon acetyl units is going to condense in the liver. This will form 4-carbon ketone molecules, and those are what are called ketone bodies. You may have heard of those. We talked a little bit. For example, beta-hydroxybutyrate and acetoacetate. Also, the molecule acetone, but it's not really used for much because it evaporates fairly easily. And so that one you don't really get a chance to do much with, other than maybe cause you a little bit of damage. So these are going to be collectively referred to as ketone bodies, and that's because they're all ketone groups. So this is going to be ketosis. We have acetyl-CoA. They're going to bond together, release 2 coenzyme A's, and that's going to give you acetoacetate. We can then process this to beta-hydroxybutyrate, and that's going to give you an NADH going to NAD+, so we'll free up an NAD+ for you to use in other pathways, though it does mean you lose some energy doing that. But you will then make the beta-hydroxybutyrate, which can go into other pathways. This is right here is acetone, so if you actually decarboxylate this, you'll get acetone. There is some uses for this pathway, but this acetone right here has the tendency to evaporate fairly easy because it's acetone like fingernail polish acetone. Okay. Now, ketosis can occur when the excessive amounts of ketone bodies is present in the body. And so because the two ketones are carboxylic acids right here, this excess formation of ketone bodies can pretty easily put you in metabolic acidosis. Acetone actually vaporizes fairly easy, so that's a hallmark of somebody that has ketosis, is they're going to have this like fruity-ish, sweet-smelling breath, kind of like you have, if you ever use fingernail polish, it'll smell a bit like fingernail polish mixed with other stuff. Okay? And so kind of a sweet, fruity, best way I can describe it. Now, the energy produced from amino acids. So we can use proteins and amino acids for energy. Amino acids are going to be a way to replenish the intermediates in the citric acid cycle. And so that's how they plug in. Amino acids like alanine contain 3 carbons. These are going to enter the pathways as typically pyruvate. Amino acids with 4 carbons typically are converted to oxaloacetate. 5 carbon amino acids are going to be converted to alpha ketoglutarate in the citric acid cycle. And then some amino acids can actually enter at more than one point. And this all depends on cellular requirements. What species do you need? What are you producing to produce other things? Amino acids typically provide only about 10% of their required energy under normal conditions. They normally, in a normally functioning human being with a normal diet, are not going to be a major energy source. Okay? They'll be a supplemental energy source. All right, now, even somebody that is like on Atkins diet, that 0 carbohydrates diets, typically majority of your energy is going to be fats, not protein. And this is actually a bit important for several reasons, but we look here, and what we see is that this is actually the different amino acids. They'll be converted to these species, which can then plug into our existing pathways at these points. Okay? So, for example, if we were to look at aspartate, it can go to fumarate right here. And then it would just go in the citric acid cycle. Okay? Notice that as long as you can plug into the citric acid cycle, you can go through gluconeogenesis from oxaloacetate to glucose. So as long as you can plug in this pathway somewhere here, you can get new glucose. Okay, and you can make your own glucose. That's why you don't have to eat any glucose. All right, so let's link it all together as best we can. Degradation of food molecules is going to begin with digestion. When the cells require energy and oxygen is plentiful, your large molecules are going to be metabolized into smaller metabolites. When the ultimately are going to be funneled into the citric acid cycle, which is the usually sort of central point that everything tends to be funneled in. Keep in mind this is in your mitochondria. This is why they play such a pivotal role in everything that you are and do. The electron transport and oxidative phosphorylation being coupled with the citric acid cycle. So through the anabolic pathways, larger molecules can be synthesized from the smaller metabolites when necessary. You can't synthesize everything, which is why you have like vitamins and minerals you need, and you need essential fatty acids and essential amino acids. The biological hydrolysis products can be shifted into the anabolic or catabolic pathways depending on the requirements of your cell. The glucose can be degraded into acetyl-CoA, which can enter the citric acid cycle. This would produce energy or can be converted to glycogen for storage in your cells. Your amino acids provide nitrogen for anabolism of nitrogen compounds. Their carbons can enter the citric acid cycle as alpha keto acids if necessary. Now, this is the entire pathways we've learned so far, all meshed along each other. You can see it gets kind of complicated once I put everything together. It looks kind of like a web. If you were to take an entire biochemistry course at the end of it, this would be like about 1/20th, 1/50th of all the pathways. So we've covered enough for you to kind of get the gist. Okay, now this is actually the end. One of the things I'd like to point out, by the way, if you have ketosis, if you're using a lot of proteins as fuel, this is actually not a good thing. because you're going to generate a lot of amine. Your kidneys are going to have to work really hard. You can also generate a lot of acetone during this process, which can damage your liver. And so you'll actually see people that are not careful with their ketosis have like liver problems, kidney problems after 10, 20 years. Okay? And you can see these people that are fitness models and should be in incredible shape, very careful maintaining their diet, their bodies, and then they like die in their 40s because they have liver conditions and kidney conditions. Okay? Very, very common, even for people that don't take steroids. So, like, talk to your doctors. If you're going to do extreme diets, don't just like do crazy stuff with your diet. Okay, it can actually cause a lot of problems. Right. Anyways, anybody have any questions? All right. Congratulations. We've gotten through. Woof. Exam coming up. Please don't forget the exam. Make sure you come. It would be a shame. That'd be bad. You all have a wonderful weekend. I won't see you until exam time, but I hope the best for you.